Thanks, Miguel, for the short introduction, and thanks to Catherine, Mikael, Daniel, and Greg for inviting me to speak about my projects, our projects in the lab. So for this presentation, I picked two of our projects that kind of gives you a nice examples to the new users, like how you can incorporate SAXS based on the need of the project. So for example, in the first project, I'm going to talk about our recent, very unique G4 DNA structure we recently crystallized. So we had SAXS validating the structure exists in cells. And the second project where I'm looking at the activity of an enzyme called MR11, where this project is like built around SAXS. So it's a different extent of SAXS usage in each project. So Albino from our lab identified this unique sequence, which is most abundantly present in the human genome. It's mostly part of the line 1 transposable element. So we'll start with that. So since I'm going to talk about a lot of DNA structure, so it might help just to recap the basics of the DNA. So since it's a SAXS-based workshop, I would like to point out when they proposed the DNA model, all they had is a very low resolution experimental data to support with. So back in 1953, there were 3 back-to-back Nature papers. In first one, which Watson and Crick authored, they proposed the DNA model where they assumed the bases would be in the keto form, and A can only base pair with T and G can only base pair with C. And also they proposed that base pairs would be like perpendicular to the fiber axis and the phosphates would be sticking out. And their model was supported by experimental data that was collected from the fiber diffraction from the DNA samples by Wilkins, Stokes, Wilson, and Franklin and Gosling. It took about 30 years to validate their model with a crystal structure that was done for this dodecamer, where they showed that Watson and Crick were right. But that being said, there are many instances where the base pairing deviates from the canonical Watson and Crick base pairing. So some of the examples are naturally occurring GG base pairing, which is called Hookstein base pairing. That's generally seen in the G4 quadrant as shown on the lower right here. So you get a base pairing between 4 Gs in the DNA, and you get this plane of G quadrat. We call it G quadruplex. So to appreciate our structure better, I would like to quickly summarize some of the G4 structures that are deposited in the PDB over years. What you see here is the G4 sequences where you have only 2 Gs in the sequence. So you could have a short sequence such as TAGG that forms an intermolecular tetramer. So you have guanines forming those quadrat I described in the previous slide. Or you could have more than 2 Gs and form this, again, intermolecular tetramer where you're getting a G quadruplex formation between 4 strands. As you increase the number of stretch of 2 Gs in the sequence, you start getting, the structures start to become more like becoming intramolecular as you increase the number of Gs. So some of the more examples of stretch of 2 Gs, if you have in higher number, all of them will form intramolecular G quadruplexes, whereas having few in the sequence, it forms preferentially intermolecular. Same thing applies to the G-rich sequences where you have stretch of 3 Gs. Like when the sequence is short enough, you get intramolecular G quadruplex formation, but as you increase the number of stretch of 3 Gs, you start getting intramolecular G4s. Especially the most studied G4 is the telomeric G4 sequence. As you can see, you have the first 3 Gs, or after that you have 2 Gs that flips out of the actually 3 TTA, they flip out of the DNA helix, and you get this bulge on the helix distortion, and then another 3 Gs that would base pair with that 3G, and the same thing would happen to another strand, and you'll form a nice duplex there. Same thing again here. I'm looking at a simple 3G rich sequence. As you can see, you have 3 Gs to base pair with another 3G wherever there is no G, that flips out of the phosphate backbone, and you get an intramolecular G4. So as you look at these sequences, there is a common trend emerge, which is if the sequence is short enough, you get intermolecular, but if the sequence is large with G-rich sequence, you start to get intramolecular G4 formation. Same thing I'm showing you here as an example for the G4 series, where you have short enough, you get intermolecular, but the moment you have adding more Gs, you get intramolecular G4 formation. For example, here that's a G sequence with 15 Gs. So again, it's the same base pairing. You have 3 Gs, G flips out, and the 3 Gs, the previous 3 Gs get stabilized by the Gs followed by that, and you get a nice stack of 3G quadruplex planes. Then what about our sequence? So this was our sequence. So if you just look at the sequence, you would predict that since it's so G rich, it should form an intramolecular G4, but to our surprise, we see a nice parallel duplex formation with our sequence. So it's almost looks like a DNA double helix, I mean the bDNA, which is the most common form of the DNA. And we have an additional G here at the 5' that gets base paired with the 5' another G. So since it's a parallel, so it's open when you have enough concentration. I mean, because in the crystal structure you have the concentration of the DNA is too high, so you would create this tetramer, which is basically head-to-head parallel tetramer. So this was our crystal structure. Since it has more similarity with the B form of DNA, so I'm just comparing the bDNA with our G422. As you can see, with this we discussed before, that the bDNA is the anti-parallel DNA with base pairs are separated by 3.5 angstrom, and the diameter of the, if you look at from the top, the helix is about 20 angstrom, and you can fit up to 10 base pairs in 34 angstrom distance. That's the pitch of the helix. Whereas our G4, we could fit almost 22 base pairs within 33 angstrom, and the diameter in one dimension is 20 angstrom. But since we have these non-G nucleotides flipping out of the helix, we get about 40 angstrom in that dimension, and the distance between base pairs still stays same as the bDNA, which is 3.1 angstrom. So, and the reason for this compactness, like it becomes so compact because of this unique sequence arrangement that's present in this sequence. So if I number these sequences in the sequence, so you have G2, G3, G4, then you have a T5 that's not G, so it flips out, and then you get base paired with G6, G7, G8, and the G9, G10, G11 continues and raises the helix, and then you have A12 which flips out, and then that the previous 3Gs get stabilized, as we have seen for other G quadruplex structures. But since these placement of these 6Gs is unique, you actually get a, instead of getting an intramolecular, you get a raise in, you get a helical formation. So if I flip that sequence, now you start seeing the Hochstein base pairing between the base pairs. To understand how the duplex formation happened in this sequence, so I just truncated this to stack of 3Gs. So I have one strand G2, G3, G4 being stabilized by G6, G7, G8, as I explained in the previous slide. And then you have one more strand in which G2, G3, G4 are stabilized by G6, G7, G8. And if you look at from the top, a single plane, you get that Hochstein base pairing I was describing in the first slide. So the closest to being a duplex G4 DNA structure in the PDB was this from BRAF promoter. But as you can see, this wasn't as helical as we have it in our sequence. And this kind of indicated as maybe if you have the 6Gs, continuation of the 6Gs in your sequence, you could actually extend the sequence and form a duplex as you would form for a bDNA. And when we look for these sequences which have placement of 6Gs next to each other separated by single nucleotide, we actually found many sequences. And one of the longest I found was G443, which is on the chromosome 12. So you have 3Gs, same as our G4, and then placement of 6Gs, and then ends with 4 more Gs at the end. So if you can imagine, based on our structure, you can easily get a duplex between the same strand sequence of this sequence. So we wanted to validate this structure in solution, so we use SAXS. So G4s are known to be very specific to, you need to have a KCl to form a G4. So first we ran the G4 G22 sequence with SAXS with water as a buffer. And as you can see, everything ends up in the void volume, so the sequence would aggregate. But the moment we ran the SAXS with KCl buffer, now you start seeing the both tetramer and dimer population. If I take the points from the dimer peak and the tetramer peak and try to fit it with the multifox as Mikhail was describing in the previous talk. I get about a population of tetramer and dimer in the tetramer peak, where you have more of a tetramer. In the dimer region, you have more of a dimer peak. So this is what we use SAXS for validating the structure in solution. And as I was telling you in the first slide, these sequences are highly abundant in the L1 line 1 element in the human genome. And one of the most active the active line 1 element in the human genome is L1HS, which has a mutation. Instead of a G, you have a C as compared to our G422 structure. So we wanted to see if this sequence folds the same way as our G4. So we did a SEC-SAX on that. Now, as previously, I get a nice separation of a tetramer and dimer peak. Now, if I try fitting with both models, I don't need to do a multi-state for this. I get 100% tetramer and 100% dimer from the peak region of those the SEC profile. So we use SAXS to validate the structure existence solution. Now the next step was to see if this duplex exists in cell. In order to do that, we wanted to develop antibody specific to that particular sequence. And in antibodies, we can have different type of antibodies. And what we went for is this VHH, which are called nanobodies. These are grown in very exotic animals such as camel, llamas, and sharks. You might have heard them because of the COVID-19. They were in news. So, but the only problem is nucleic acids are generally very weak they're not a good immunogen. So using a live animal is not an option. So we contacted this company called Hybridgenics in France. They have a pre-made library of 1 billion clones of nanobodies. So we use them to screen for binders that would bind specifically to our sequence versus a duplex sequence. So in order to do that, I wanted to form a duplex, but instead of just using random duplex, since I wanted to be specific, so I wanted to take our G4 and make a complementary of it and try to form a duplex. And in order to test whether it's forming a duplex or still forming a G4 sequence as seen in the crystal structure, the assay I developed was, what I found was MRE11, which you will hear in the second part of the talk. It's a nuclease, it cuts the double-stranded DNA. But what I found was when I do the nuclease reaction on the G4 sequence, it doesn't cut. So here I'm showing you the nuclease reaction. So here you have a label on the 3' end of the sequence. So when you form a duplex, G4 duplex with that sequence, you get a 3' label on the 3' ends. And MR11 cuts from 3' to 5' on a double-stranded DNA. So here the first line is substrate only, and then I have a MR11 wild type in the presence of manganese, because nucleus is a manganese dependent nucleus. And the third lane is basically no manganese, and the fourth one is a nucleus dead version. So what we are seeing is if the MR11 is active on this substrate, it should cut the tag on the 3' end, and you should get a product somewhere here. But for just the G4 by itself, when you don't have a complementary sequence, I don't get any product. So that means MR11 can't cut this substrate. So G4 is not a good substrate for MR11. But if I incubate G4 with complementary sequence, boil it and cool it down in the presence of a complementary sequence in excess, I do get a little bit of product, but mostly MR11 doesn't cut it, which tells me that the duplex formation in this assay is not feasible. So in order to achieve a proper duplex, what I did is basically connected the complementary sequence with a T4 loop to the G4 sequence. Then you have two possibilities. One, you could end up G4 preferentially forming between two strands, and you would get the complementary strand as a single strand DNA. And if you do a reaction with MR11, you wouldn't get the product. Or you would have a second case where you actually would get a intramolecular duplex formation, which would be a good substrate for MR11, and you should start seeing the product. And that's what happened when I boil this substrate and pull it down. You see most of the substrate is in duplex formation. So we got a nice duplex as a negative control for selecting nanobodies that would bind to G4 versus the similar looking duplex. So we use those two. For positive selection, we're using biotinylated G4 sequence. For negative selection for the nanobody, we were using the T4 loop duplex with a biotin tag. So here we are expressing nanobodies on the phage and doing a phage display to select the nanobodies that are specifically binding to the, first you'll pass those phages through the negative selection antigen, which would remove everything that would non-specifically bind to either duplex or the solid support. Then you would take the rest of them and pass it through the positive antigen, and you will be able to get the binders that are binding to your sequence. We did that, and we have about 10 nanobodies now we are testing, which are specifically binding to the G4 sequence compared to the duplex sequence. So we are hoping to use this to prove that this structure can actually exist in cells, either with ChIP-seq or immunofluorescence. So this was the first part of the first project. So now I'm moving to the second project, where we are using SAXS as a readout for measuring the activity of this MR11 nucleus. MR11 is a part of this complex called MRN, which consists of RAD50 and NBS1. RAD50 is an ATPase, and NBS1 is an adapter protein that binds to phosphorylated proteins. So MR11 is known to cut the double-stranded DNA and would make this 3' overhangs for break to the DNA, the double-stranded break to repair. Yeah.
Five minutes.
Okay, I'll finish it, because I think Daniel is going to talk more about this project, so I'll let him speak more about the technicality. So I'll just summarize what we did here. And for this, we wanted to use SAXS based on gold DNA substrate, because I mean, by now you might have already realized that SAXS is a contrast measurement, and there is a very little difference between the electron density between water, protein, and DNA. So the gold nanoparticles kind of gives you an advantage where you're going to actually get a huge boost in the signal, as shown in this figure here, extracted from this PNAS paper by Greg, where you're basically, by measuring the correlation scattering function, you can actually accurately measure the distance between the gold label that are connected to the DNA substrate. So Greg, this has been done previously, and they were able to accurately measure the distance between two gold labels on the DNA. Greg used this method in his 2013 PNAS paper to see how this mismatch repair protein bends the DNA based on whether you have a mismatch or the normal duplex. So we wanted to do a little bit different, since MR11 not only just binds to DNA, but it cuts the DNA. So we wanted to design substrates such that at the end of the reaction, you would have two gold nanoparticles separated from each other. So first, we validated those substrate in our gel-based assays, where instead of gold nanoparticles, I have fluorescent labels. So MR11 would cut those substrate, and you get the products. So I'm just going to shoot to the model. So what we are trying to do here is, when you build a PFR from this gold-based assay, you get two peaks, PFR distribution, one corresponding to intra-gold distances and one for the inter-gold distance. What we are trying to do is after MR11 nuclease activity, you should see decrease in the amplitude of the PFR for the intergold, where you're normalizing your data to intragold to account for the changes in the concentration of the gold nanoparticles in your experiment. So Daniel from SIBYLS made these substrates. And as you can see, when he normalized the intragold, these are the substrate with different lengths. You could actually pick out the difference in the length in the intergold of peak of the PFR. So when we actually did the experiment with, this was like after a lot of optimization, a lot of customized SAXS experiments. So this has all been done with high throughput SAXS. So we are looking at 6 population here, where you either have just the DNA, or the DNA with wild type, no manganese present, so that the enzyme is inactive. And then you have a nucleus dead, again, no manganese, or you could have a DNA substrate, just with the manganese, and now you have a complete reaction mixture, where you have a gold labeled DNA with wild type with manganese, so you should see the nuclease activity. And then you have a substrate with nucleus dead in the presence of manganese chloride. So in the 3 cases where you have just the enzyme, either wild type or mutant, nucleus dead mutant, or the nucleus dead mutant with manganese chloride, you should see the protein binding to the DNA changes the intra-gold distance. So you should see it as a changes in the PFR. But when you actually have the wild type with all the manganese chloride, you actually see the reaction, and then you start seeing the decrease in the amplitude of the PFR for the intergold distance. When you don't have any enzyme, you just see the distribution for the free substrate in the absence of enzyme. So to summarize, we use SAXS and crystallography to study a unique G4 sequence from the human genome. And our structure indicates that this structure could be extended if you have a proper arrangement of those 6 Gs in the sequence. And also we showed that with high throughput SAXS, with a suitable DNA substrate, you could actually not only structure, but you could actually get a readout on the activity of an enzyme. So with that, I would like to conclude my talk, and I would like to thank everybody in the Trainer Lab who are part of the G4 project, as well as Susan at LBNL and Andy Arwai at Scripps. And big shout out to Daniel for the second project where, as I said, he was very involved in this project. So we published our essay as a method paper in Methods in Molecular Biology. So if you want to learn more about it or to see what are the tips and tricks, I would refer to this paper. And also thanks to Rob and Sally Group for Scatter and Fox web server, and of course the funding. And thank you all for your attention. And I would be happy to answer any questions you may have.